All right, so as she said, my name is Emily Boyd. I'm from Regina High School. And this summer I was given the project of working with ovarian cancer and understanding M6A modification. So as many know, ovarian cancer is a very deadly disease. In 2020, there were an estimated case amount of about 22,000. And in 2010 to 2016, less than 50,000 survived from it, while 80% within 5 years reoccurred. And overall, ovarian cancer is going to be characterized by metastases, which is what I'm focusing on in my project. And what causes metastases is epithelial to mesenchymal transition. So as we can see, an epithelial cell is going to be a normal cuboidal cell. And as it transitions, it's going to become a spindled cell that is able to migrate and invade and form tumors. So recently, EMT has been found to work with epigenetics. And as we can see, as an epithelial cell is becoming mesenchymal, it has the option to undergo epigenetic modifications. And for my project, I'm looking at RNA methylation. So RNA methylation is a post-transcriptional level of regulation. It's simply just the addition of a methyl group on an RNA. So in my project, I'm looking at M6A, which is just going to be an adenosine base with a methyl group added at the nitrogen 6 position. So M6A is an abundant eternal modification found in mRNA. It entails, as I said, methylation of adenosine base at nitrogen 6 position, and studies have demonstrated that M6A is reversible, and it is key to regulation of the RNA metabolism. So for this to occur, we have M6A writers. So these are going to be proteins that induce the methyl group. This can be METL3, METL14, or METL16. And then to demethylate, we're going to have proteins that remove the methyl group. These are considered M6A erasers, and that is FTO or ALKBH5. And then once methylation and demethylation has occurred, readers are going to come through, read the RNA codings, and carry out whatever has been coded for. So that can be like pre-mRNA splicing, RNA decay, or RNA stabilization. So this whole process has been indicated that EMT and epigenetics are important in cancer development. However, it hasn't been shown in ovarian cancer. So our objective is to investigate whether mesenchymal ovarian cancer cells have higher level of M6A compared to epithelial cells and its role in EMT. So our first hypothesis is whether mesenchymal ovarian cancer cells have higher M6A RNA modification than epithelial cells, which we're assuming will have higher writer and lower eraser. So our first question of our first experiment is, do mesenchymal cells have higher M6A RNA modification compared to epithelial cells? So to test this, we're going to conduct an RNA dot blot. To do this, we're going to do one epithelial cell line, R182, and two mesenchymal cell lines, F2 and R182. So to conduct the dot blot, we start with growing cells and feeding them. We're going to lyse the cells, separate the cells with chloroform, isolate the RNA and extract the pellet, and then suspend it in water to have RNA. And then with that RNA, we're going to dot it onto our membrane with 100 nanograms per dot. Expose it to UV to link the RNA to our membrane, and then incubate it in a primary and secondary antibody conjugated with HRP. And then we'll image it. So when we did this, this was our image. As we can see on the left, our epithelial cell, it's much lighter than our right. That means there's not as much M6A present as in mesenchymal cells. So we can conclude that with this, mesenchymal cells have higher M6A than epithelial cells. And then our second question, we're looking at if mesenchymal cells express higher M6A writers compared to epithelial cells. So our hypothesis is, do mesenchymal ovarian cancer cells have higher M6A writers compared to epithelial cells? And to test this, we're going to use qPCR to quantify the RNA expression level of M6A writers in our different cell lines. So for epithelial cell lines, we're using R182, R2615. And then for mesenchymal, we're doing F2, SKOV3, and OVCA433. So the writers we're going to be testing is METL3, which methylates RNA, METL14, which enhances METL3 binding, WTAP, which stabilizes METL3 and METL14. Those three are going to be found in the methyltransferase complex. They're going to work together. And then METL16 is an outside writer that methylates RNA and non-coding RNA. So when we conducted this, we can see METL3 using our epithelial cells as our control. Mesenchymal cell line F2 has almost 30 times more METL3 show up than both epithelial cell lines. And then METL14 has about 5 times more METL14 show up than our epithelial cell lines. And this is proving methylation is occurring because in the complex, they're bound together. And then WTAP, there's about 2 times more F2 shown than R182 and R2615. And then METL16, F2 has about 10 times more METL16 shown up than R182. So we can conclude that mesenchymal ovarian cancer cells do have higher expression of M6A writers when compared to epithelial cells. And then our last hypothesis is, do mesenchymal cells have lower level of M6A eraser compared to epithelial cells? So we're asking if the expression is lower of M6A eraser. So to test this, we're going to do qPCR again with the same cell lines, just different erasers. And then our result when using FTO, we see that FTO is demethylating more compared to mesenchymal cells, which validates our other research. And our conclusion is that mesenchymal ovarian cancer cells have lower expression of eraser compared to epithelial cells. So in summary, our mesenchymal cells have higher M6A modification compared to epithelial cells. This is supported by the higher expression of M6A writer showed in our qPCR, and that FTO is lower in mesenchymal cells compared to epithelial cells. And this data highlights the importance of further investigation on the role of EMT in ovarian cancer and allows us to inhibit ovarian cancer metastases. So in the future, we would prefer to immunoprecipitate M6A and sequence the RNA in the downstream genes that control M6A modification. So basically, we would fragment that RNA, expose it to M6A, and then get that sequenced and understand what other genes we need to be looking at to inhibit how the metastases are occurring. And then, thank you.